Welcome to Physical Chemistry. There has been a recent development in science that is interesting to us in our particular topic. At exactly this point, and it happened or at least it was publicized in the news just recently. It is a blue dye that can be made from red cabbage, that can be used to make disgusting blue ice cream that weird German children seem to like. It's actually typically vanilla flavored, but it's bright blue, and it uses a coloring blue FCF, which is questionable. You may also have heard, those of you that are international students particularly, you may also have heard it's quite unusual that, for example, some types of American sweets that are bright blue are not allowed to enter Europe, so you can buy the same things like Skittles, M&Ms and things, but the blue ones are taken out. Obviously they don't put them in the bag to start with, but there is a different version sold in some countries than others, and it's all about this slightly toxic and not very well sourced blue dye that comes from oil, effectively. So there is derived from a vegetable alternative that is bright blue, and it's very recent, and it was a big news. Why does it relate to us? Well, it's a pigment that comes from red cabbage that's typically a purple colour. And it's a purple colour because it's a big molecule. So the double bonds, there are a lot of double bonds in there that cover the whole molecule, and they can have a lot of energy states because we can have lots of possible orbital states inside our long double bond. So we've got not only the bonding and anti-bonding of one end. If we've got one double bond, if we've got another double bond to it, we've got the bonding and anti-bonding of that, and so we've got 4 states, and they get closer and closer as we get more and more and more, and so the absorbance gets redder and redder. So if we take a molecule and make it very big, then we would expect that it would absorb in the red end of the spectrum and appear to be blue. But there's a typical problem that if we make the molecule too big, then it absorbs too much and it doesn't look bright blue to us. So that's the normal problem that we see, and that's why there aren't very many bright blues in nature. The typical butterfly and insect color comes from a different technology comes from a different evolution solution of getting the light and sorting out the colors that they want and what they don't want. It comes from a different physical process than absorption, which is what we want. So in this particular molecule that we're interested in, the researchers have taken a natural plant molecule and changed it slightly. Here is a picture from the actual paper. The paper is in Science, and it shows 3 of the molecules attached to an aluminium center. So we've got the aluminium in the center of the picture. On the left we've got one way of showing it, and on the right there's a different way of just drawing the same molecule. Let's look at the one on the left because it's got the critical part of the molecule in blue. So if we look at it on the left, we can see, we just choose the one on the top left as an example. We can affect the angle of this bond, these 3 atoms to each other, by making the sugar on the end, which is this piece here, we can make that bigger or smaller, and that forces the molecule to twist a little bit so that it can fit onto the aluminium. The oxygen to aluminium bonds are very strong, it's charge transfer, so it's a very, very strong interaction, and that can force some of the other bonds out of position, and that will change which double bonds interact with which other ones, because although they are a double single bond situation with all sp2 hybridized carbons, so we could theoretically have a completely conjugated system and a very long wavelength absorption. We can modify it by twisting the bond, which isolates one part of the molecule from the other. It changes the energy, and we can see in this little graph here that as we twist the bond more and more, what we see is a bluer and bluer apparent absorption. It's not in this case because the actual wavelength is shifting, it's because the shoulder to red is actually decreasing, and that's because we're changing the interaction of the two parts of the molecule with each other. It's a little bit more complicated than what we're considering, because it doesn't break it into two parts that absorb both in the visible region, because we get out of the visible region completely, and the aluminium is also interacting with the molecule and affecting the color. But if we take different substituents and cause it to rotate different amounts, we can change the apparent color of this molecule just by changing the energy states of those different possibilities for the electrons inside that long double bond. So this is a new research, because now we can make a completely natural and natural-ish molecule. Effectively, they've taken a dye from cabbage, which is a purple color, and grafted on a sugar using an enzyme, and now it is a bright blue color instead of a purple color, to us at least, and that causes it's a rare color for nature, and therefore useful for the food industry, because then you can make blue food, which is particularly shocking and interesting, and it's otherwise very difficult to do. There isn't part of the reason why blue food is shocking is because it's rare, and that is why it's interesting.